Cells in our body face an enormous challenge. They have to fit all our genetic material, the DNA, into a tiny space. It's as if you would try to fit 40 kilometers of fine thread into a container the size of a tennis ball. DNA can be found in the cell like an intertwined set of strings, all stuffed into a dedicated compartment. When a cell divides, it needs to split and evenly divide the DNA over the resulting daughter cells, which is quite an achievement. Let's say DNA is like your Christmas lights that you need to untangle after having been in the box for yet another year. You can't just start pulling and hope that they come apart. You have to organize them into individual strings first, then wrap them up and make sure you have a handle on them to pull them apart carefully. In the case of cells, we know the individual strings as chromosomes. When you look at a single chromosome, what you can observe is that when the cell is about to divide, the chromosome condenses, is brought to the middle, and then pulled apart. Importantly, the pulling should only happen when the DNA is fully condensed. The microtubules are the structures in the cell that do the actual pulling. Perhaps think of these as ropes that attach onto specific sites of the DNA to pull. When you now zoom in on one of these sites, you will find a machinery of proteins that all help to create these binding sites on the DNA. We call these places the centromere. In our research performed in microscopic worms, we focused on one of the main players that helps build the centromere, KNL-2. What we found is that this key player not only builds the centromere, but, at the same time, is also directly involved in condensing the DNA. So two distinct roles, both crucial to make sure DNA is able to segregate well. To go back to the Christmas light analogy, KNL-2 turns out to be the player that helps to neatly wrap and organize the lights, while at the same time also creating a handle to pull them apart without breaking anything. So how can one protein have multiple functions, you may ask? In this case, what is important to know is that proteins can be modified, which can alter their function. These modifications are highly dynamic, meaning that they function almost like a switch, switching a protein from having one function to another. We discovered that KNL-2 is also being modified and found the exact spots on the protein where this is taking place. To understand the impact of this modification on the functioning of KNL-2, we decided to change this part of the protein so that the modification could no longer occur. We then took a closer look at a dividing worm cell to see how it would cope with this small change. Things looked really bad. Whereas, as you've seen earlier, the chromosomes normally condense until they're compact enough to be pulled apart correctly, without this modification on KNL-2, we could see that the condensation didn't go to plan. The DNA clearly wasn't condensed enough when it needed to be pulled apart, which led to the missegregation of the DNA. Interestingly, preventing the modification on KNL-2 did not have an effect on the centromere. These were still built in the right places. Preventing this modification specifically affected the proper condensation of the DNA. This leads us to this final picture showing our current understanding of KNL-2 functioning in dividing worm cells. It plays a role in building the centromere and, specifically when it is modified, also helps DNA to condense enough before it is being pulled apart. So when you're putting up your Christmas tree this year and get frustrated by the entangled Christmas lights, be inspired by KNL-2, who in worms takes care of a similar problem each cell division. Quite an astonishing feat if you think about it.